All right, so we're going to start by doing this little drawing of the fluid mosaic model. I have there the players in this model. And the first is a phospholipid, right? So a phospholipid is interesting because it has a hydrophilic head and hydrophobic tails. And we know that hydrophilic and hydrophobic don't or repel each other. And so because of this structure, it allows us to have a lipid bilayer. Okay? I'm going to draw a little space between these because we're going to draw these other things in between them. All right, so on the inside of the cell is the cytosol, which is mostly water. And the outside of the cell is generally water as well. So that's where the hydrophilic heads point and the hydrophobic tails point towards each other. Okay. Cholesterol. So we often hear about cholesterol when talking about artery diseases or artery function. But cholesterol is found in all of your cells, every cell. And I'm not going to draw the full structure. It's a bunch of rings, but essentially creates this hydrophobic ring structure. And then a little hydrophilic OH on the end. And so it fits in our fluid mosaic model in between these phospholipids, okay? And it creates space, essentially. Breaks up, if there's not enough cholesterol, all these tails will attract towards each other and make it too dense, but the structure of cholesterol is a little wider and breaks them apart, and so it allows for more movement and flexibility within the plasma membrane. All right, this is annoying. So our P, what did the first P stand for in P-PIMP? Peripheral, right? So we've got a protein here. Proteins are going to have hydrophobic and hydrophilic regions, and those are going to line up then within that lipid bilayer according to those same properties. Okay, but peripheral means it's not very big or not very wide, so it's not going to span the whole lipid membrane. We'll stay on the periphery. It could be on the outside, or we could put another one over here on the inside. They may even shuttle between. Okay, but they don't span the whole lipid bilayer. As opposed to, I'll use a different color. Our integral plasma membrane proteins, which are much larger, and they are going to span the whole lipid bilayer. And these make great channels, okay, for things to get in and out of the the cell. The peripheral and integral ones may be receptors, they may be enzymes. There are a number of functions in the list from the video lecture. Okay, and some of them may have the same function, but may do it slightly differently. All right, what's a glycolipid? If you break those apart, what does that mean? What?
Glycogen and the lipid, it's sugar fat.
Okay, good. So where's the lipid part going to go?
Tail.
All right, so we'll give it a red tail. And it's going to have basically a string of sugar molecules on it. Okay, and those sugar molecules will probably be outside or inside the cell. A lipid stuck in there. Okay, and these are usually for cell identity, so they help identify what type of cell it is. Okay, and similarly, a glycoprotein is going to have a protein section and a string of sugars on it. All right, the only thing missing from here is we have also parts of the cytoskeleton, which are going to embed into the lipid bilayer as well. Okay, so this is intermediate filaments and microfilaments, which are going to help stretch from one part of the cell to the other. They may carry things like railroad tracks, or they may just be for maintaining the shape. Okay, and where they then insert into there, it's called a picket fence.
So the space between the phospholipids, those are called picket fences?
No, the places where the cytoskeleton goes into the lipid bilayer would be the picket fence. Yes?
Are there picket fences on both sides, so it's from the cytosol as well? Or just from
No, just from the side, just from inside the cell. Yeah, not from outside the cell. All right, so this is our fluid mosaic model. Fluid means these aren't all permanently in one place fixed, okay? These things can move throughout the cell, and the cholesterol allows it to do that. It breaks up the lipids, so there is some movement allowed in there, okay? Mosaic, meaning there's a lot of different things on one page, right? Or a lot of different things come together to form a functional group. All right, any other questions? The fluid mosaic model, okay. All right, let's move on into the cell. And the cell has three basic features. And what are they? Why don't you talk to your neighbor so you can name all three? Well, we just talked about one of them. What was that? All right, good. Our plasma membrane or cell membrane. Okay. We talked about one part that connects into and also goes inside, right? The cytosol and cytoskeleton. And the third one?
Organelles.
Okay, good. Organelles. All right, now I know most of you have done this before, but let's see what you remember. Okay, I want you to draw all the organelles in your little cell. Okay, I'll give you a few minutes to do that. All of them. Okay? But there's the figure in the book. It's on the worksheet. Okay, so know that figure. It has a little more detail than this. And then the functions of these you should know as listed in the video lecture. Okay? We can go over the ones that I have listed. All right, so what's the function of the ribosomes? Okay, all right, so that's where proteins are made. Okay, which is fitting that it's right by the nucleus because what does the nucleus have in it? DNA. So those are the instructions for building proteins. So you can go make your RNA. RNA goes to the ribosomes. You make your proteins all right there. There are some free ribosomes, however, which are just in the cytosol, which I also don't have labeled. Okay, but some of them reside on this endoplasmic reticulum. So if it has ribosomes, it's rough, and if it doesn't, it is smooth. Okay, and the endoplasmic reticulum also can be for storage. It can be for packaging things and sending them somewhere else. It also, some of them have the ribosomes on it, so they can be for making proteins. All right, vacuole. Not a lot of cells have these, but when they are there, they're for storage. Okay, plants have big vacuoles. Most animal cells don't have them. All right, the Golgi apparatus, it makes these vesicles. Okay, so it basically makes a little transporter for whatever may be in there. So you could have a protein in there, you could have lipids, and it keeps it within there. You can also, this is where lysosomes and peroxisomes are going to be made, or at least the vesicle part of it. Lysosomes are for digesting old and damaged organelles. Peroxisomes are for counteracting the free radicals in hydrogen peroxide and other byproducts of cellular respiration. Cytoskeleton, we talked about that. It maintains the shape of the cell. It can also help transport things throughout the cell. And then the mitochondria is the powerhouse of the cell, as we all know, where cellular respiration takes place. Okay, so you guys got this. This is basic biology. But we'll refer to this when we talk about different tissues and cell types and systems and what type of cells are found in different areas. You know, some may be mitochondria rich because we need a lot of energy available in that location. Others might have, such as muscle cells, have a sarcoplasmic reticulum, which is a modified endoplasmic reticulum for calcium storage. So these will show up again and again as we go through our tissue systems and cells. Okay, any questions on this? Okay. All right, let's talk about electrochemical equilibrium. Did anyone find this confusing? Okay, all right, good. That means you were trying to learn it, okay? Because it is confusing, but we're going to go through it kind of a step-by-step fashion, and hopefully it'll make sense. So if you start with, we have our cell here, and it's going to, what if I just hit allow? All right, it's going to have certain amount of ions in greater concentration inside or outside the cell. And we've got negatively charged ions and positively charged ions. These aren't all the ions, but these are the major contributors to the difference in charge. That's what we're talking about with the electrical part. Difference in charge inside and out the cell. Inside and outside of the cell. Now, this stands for proteins. Proteins are not ions, but we're just throwing them in there because they also affect the charge of the cell. And the proteins affect the charge more than anything else. Okay, and these are big negatively charged proteins. So we're going to do a big negatively charged protein inside of the cell. Now, because they're big, what does that mean they can't do? Yeah, they can't leave. Okay, they're stuck inside. So already we have a precedent for having a negatively charged cell. So we have these bunch of these proteins in there, and they're negatively charged, and they can't leave. Okay. The other player that is in great concentration inside the cell is potassium. Okay, so there's a lot of potassium inside the cell. And because it's a positively charged ion, it is attracted to the negatively charged proteins. Okay? So all of the positively charged ions will be attracted to anything negatively charged, and vice versa. Okay, but what we're trying to show here is the point at which the charge is in equilibrium with the concentration. Okay, the concentration gradient, I mean. So we know that things diffuse from greater to lesser concentration. But proteins can't diffuse in this situation. So we'll draw proteins. We'll draw a little small one outside and a little small potassium outside, showing they are in concentration, but in small concentration outside the cell. All right, these other ions are all in greater concentration outside the cell. And in smaller concentration okay, calcium and chlorine will come into play when we start talking about heart cells, okay? But we're going to ignore them for now and just assume they're going to stay in place. But sodium and potassium play the biggest role in changes in charge inside the cell. So we've got three players here, three integral plasma membrane proteins, which act as channels or transporters of ions. Okay, so we'll first talk about, all right, we've got sodium in green and potassium in red. So we're going to make a red channel to represent our leaky channel, our leaky potassium channel. So if you put a little channel here, these are going to be around the cell, and you ignore the charge, what is, so according to the concentration gradient, what is potassium going to want to do? Exactly, leave. So it's going to leak out of these cells.
Those are called leaky channels.
Yeah, these are our leaky potassium channels.
They're also called sodium potassium.
Nope, we'll get to those. All right, so we've got leaky potassium cell channels as well. And which way is the sodium going to leak through? Into the cell. All right, so the point at which the electrical charges are balanced out by the leaky channels, by the diffusion gradient, gives you an overall negative charge of 70 millivolts inside the cell, if you're just looking at the leaky channels. Sorry, that's wrong. 90. All right, so far we've just talked about passive processes. But we also have these sodium-potassium pumps. And they are going to pump sodium, potassium inside and outside the cell.
It's not like because the sodium leaky channels are not as permeable as the potassium, more potassium is going to leave than sodium goes in. That's why it's not like a one-to-one exchange. Otherwise, wouldn't the net charge be the net membrane charge would be 0?
Well, I'm not sure exactly on the leakage of those channels. It's a good question.
Negative 90 millivolts kind of represents kind of resting potential that's happening
Right.
inside the cell. So that's just there being happy content. So when an electron in that or neuron passes by, that's when your sodium potassium channels open up, and as we hit the negative 70, that's where all the changes becomes.
Yeah, so at this point, if you ignored the sodium potassium pump, which is working, which we'll show is going to be 2 for 3,
Right.
that's going to create
So then that negative 90 just comes from
the proteins.
the high amount of proteins that are negative, even if there is a one-to-one exchange of potassium.
Yeah, so it's
Right.
just the resting potential of the cell. It's just chilling at the negative 90, all is good.
Right. Okay. But yeah, you don't have to know the details. Just know if you just look at the leaky channels, the proteins, potassium and sodium, you're going to rest at negative 90. Okay. But the sodium and potassium pumps, okay, these guys use ATP. So it's going to be an active process. And they're going to pump sodium and potassium against their concentration gradient. So that means they're going to pump potassium in. Okay, while at the same time pumping sodium out. Yep.
So it's like a sump pump, basically.
Yeah, this takes work to do it because you have got to get it out, got to go against gravity. They're essentially going against their concentration gradient. All right, and it's going to change this. It's going to make it actually less negative and more positive. And the reason it does that is because for every 3 potassiums it pumps in, it only pumps 2 sodiums out. So with the sodium-potassium pumps, you then settle on a negative 70 millivolts for your overall net charge at rest. Okay, does that make sense? Does that make more sense, I should say?
So the leaky channels are negative 90 millivolts and the negative 70 are the pumps.
Right, so after these sodium-potassium pumps go to work, they're going to make it less negative, and you're going to rest at negative 70. Okay, and this is cell-specific. So most cells are at negative 70. We look at muscle cells, or we look at neurons, they're going to be different.
So, but both of those are a negative number? The 70
Yes.
and 90? Okay.
Yeah, so the negatively charged proteins are still carrying most of the weight. They're still going to make it overall negative. But we can make it less negative by using the sodium-potassium pumps. Okay, any other questions? Yes? Well, it maintains this electrochemical equilibrium, but it is an ion used for neuronal stimulation. So when these change in great frequency, and we'll talk about this in the nervous system, it's how we send a signal down or into a cell. Yeah.
I was just going to ask why it's important that it be at negative 70 and not 90.
Right. Yeah. So I don't know why certain cells are different, but they are. And then these aren't static, so these will change. But this is all we're going to learn for now, because this will give us a base. So when we talk about like muscle stimulation or neuronal stimulation, we're going to start here, and then we're going to pick up how it changes. Yes, Allie? Just doing your hair? Anyone else? Okay. Yeah. So the leaky channels, what type of passive transport is that? be facilitated diffusion, right? So they're going through a channel from greater to lower concentration. But yeah, that would be a passive transport, facilitated diffusion. Okay, what about the sodium potassium pumps? It's active. What type? Yeah, it's an antiport. So you have two ions using energy going in opposite direction at the same time. Well, it's not actually at the same time, but yes, antiport. All right, so a good study thing to do is to maybe duplicate this slide, erase everything, and then go through it just like I did. All right, start with the proteins, start with the potassium, then the relative concentration of the other ions. Put in the leaky channels, what's that going to do? Put in the sodium potassium pump, what's that going to do? Yes?
I mean, the main purpose is to know what are the functions, not necessarily how it looks like, correct?
Yeah.
Because that's how you would test us on the exam.
Yeah, so there's a figure on the worksheet. It has, I wouldn't say a better drawing. Yeah. Well, not that one. I think it's another one. That one, yes. And it has all the things on there. Okay, any other questions on this? So again, we're looking at the electrical charges and then the chemical diffusion rates, essentially, of different ions and our big negatively charged proteins. Yes?
Is the negative 90 millivolts in the cell, is that like taking into account the protein's negative? Because in the video lecture it talks about how it would like hold some of the potassium in the cell because it's a negative charge. Is that like before or after that attraction's been taken in?
So all these forces are taking place at the same time. But yes, after you account for attraction between ions, that's what we're at.
So if there were no attraction between like the proteins and the potassium, then the number would be even more negative?
Right, yeah, I guess. Well, it also, yeah, if it was a neutral ion, if there's such a thing.
Then it would be negative because
Yeah.
the protein would connect,
But yeah, this is the way it is. And it's the only way it works. So, yeah. I don't know exactly why. But this is what it is. All right. Any other questions? Okay, let's talk about or review these active and passive transports. We already mentioned a couple. These are definitely important when talking about cellular processes. So, for example, digestion and absorption. How do we break down sugar and then how do we get it into our bloodstream? It's going to depend on these active and passive transport processes. We just talked about electrochemical equilibrium, again, based on active and passive transport. Okay, so what are our 4 passive transport? What's the simplest one? The simplest one is diffusion, right? And diffusion with water through a semi-permeable membrane. Okay, and then through a channel. All right, then what's the last one?
Bulk flow.
Okay, bulk flow or filtration. Okay, and this is constantly occurring through your capillaries. The force from your heart is forcing fluid through your capillaries into your cells. All right, active transport. Yeah.
I thought blood flow was part of water transportation because there's a difference in pressures.
So what's your question?
So bulk transportation is where there's a difference in pressures, like something is having to suck or push to another area.
Yeah, so that doesn't fall under our passive. Bulk flow, I think is what you're thinking of.
No, that's not.
All right, well, I'm not sure what you're thinking of. But it is because of pressure that this works, right? The pressure from the heart forces fluid through. But at the site of the capillaries, there's no energy required. It's already been done. So our example from last year, evapotranspiration is another example of bulk flow. It is energy that's fueling the process, but at the site, it's just being pulled or pushed. Yeah. Okay, our active transports, we have vesicular transport, of which there are a few different kinds. All right, we've got phagocytosis, and pinocytosis. Oh, didn't work.
Did use the bulk transport though, term and slash.
Oh, bulk flow, bulk. Oh, you're talking about which one? Vesicular? Yeah. Yeah, bulk transport. You can say that as well. All right, what are some other active transports? We have. What's that?
Primary active.
Okay, primary active. Okay, what does that mean? Well, you're using ATP to move one thing in one direction. One D? Yeah.
So I thought primary active, it would like go against the flow.
It is.
I mean,
It's still going against the concentration gradient, but just one thing at a time.
okay.
And then secondary active is two things. In either the same co or sim transport. I've tried them all. Or opposite direction. Yeah.
So the sodium potassium pump would be an example of secondary active.
Secondary active, antiport. Yeah. All right, so again, when we're talking about cellular processes, how substances move through our plasma membranes, we're going to go back to these again and again. And so this will show up over and over. All right, any other questions? That's all we got for the lecture.